Hello, my name is Shawn King, and I am an undergraduate student at Arkansas State University. I am a biological science major, and I had the pleasure of participating in the Arkansas Biosciences Institute internship in the summer of 2019 with Dr. Neuman-Lee and her lab. I have continued to work in the lab, my focus being on my work to examine blood samples from radiated sliders by making smears and observing them under the microscope for immune cell morphology and relative proportions. Red-eared sliders are turtles that are common in northeastern Arkansas. They are found in many habitats, but we examine them in water surrounding agricultural farmlands. In the wild, turtles are exposed to many kinds of stress and infections such as endoparasites. One endoparasite is the hemoparasite Plasmodium. These blood parasites are visible in blood smears. Mosquitoes are their primary vector. In addition to examining parasites, blood smears allow us to examine important immune system cells, or white blood cells, such as eosinophils, which are high in times of parasite infection, heterophils, which are phagocytic cells, and lymphocytes, which are a second line of defense in their cells. Turtle immunology is not very well known, so studying basic cell biology has huge benefits to understanding disease and immune function in turtles. The main goal of this project was to get a baseline immune cell proportions for red-eared sliders by determining their heterophil lymphocyte ratio, also called HL, or HLR in my poster, eosinophil count, and measuring Plasmodium occurrence. Blood samples were collected at intervals of baseline, 15 minutes and 30 minutes from the caudal vein and the tail in the summer months of 2019 in Beritage Farms, Cherry Valley, Arkansas. Blood film slides were taken and smeared, then fixed in methanol. Smears fixed were then taken to the lab to be stained in James Wright stain for 1 minute stain, 1 minute buffer, and 10 dips of rinse. Slides were then dried overnight. Slides were examined under the Nikon 1200 light microscope for observation of red blood cells and white blood cells. I noted any Plasmodium in the red blood cells and counted eosinophils, heterophils, and lymphocytes. I would like to refer your attention to the figure on the right. The x-axis has heterophil lymphocyte ratio, and the y-axis has the number of Plasmodium. There was no relationship between Plasmodium and HL ratio in the red-eared sliders sampled. The graph in the middle, the x-axis has the number of eosinophils graphed, and the y-axis has the number of Plasmodium. We found no relationship between Plasmodium and eosinophil counts. The graph on the left, the x-axis is sex, the y-axis is HL ratio. We found no difference in HL ratio between male and female turtles. HL ratio is typically indicative of higher stress, so we expected to find a difference between the female and male HL ratio because female turtles expend energy on egg production during the mating season, which has during the sampling period we took. We also expected that all turtles would have a higher HL ratio and a higher Plasmodium load. We did not see either of these relationships. It is possible that other infections could have been present. There are other stressors that we did not measure. We also predicted a higher level of eosinophils in turtles with higher levels of Plasmodium. In mammals, eosinophils function as antiparasitic cells, but their role may be different in reptiles. Additionally, different seasons could influence Plasmodium occurrence since mosquitoes are more active during the summer months, which was when the samples were collected. Finally, we don't know what these turtles have experienced prior to capture. The environment of other stressors and infections could have influenced the results when blood was sampled. Future work could be determining Plasmodium species present and increasing the sample size to include more individuals. With that, I am very thankful to my mentors, Dr. Neuman-Lee and Benjamin Spitz. I am also very grateful to the help from the members of the Neuman-Lee lab. Finally, I am so grateful for the opportunity to do this work through the ABI internship at Arkansas State University.